Hi, have you ever wondered why we all look differently? So, have you checked yourself in the mirror and wondered why you don't look like your parents or even your siblings? The answer to that is because all of us has characteristics that are unique to each individual. Aside from humans, animals like dogs may also exhibit this variety of characteristics. So, like the different breeds of dogs, you can see that they are larger dogs, they have different physical features. Although these dogs look different, the different breeds can mate and have viable offspring, and with that, we can consider them belonging to the same species. In plants, we could also see here in peppers that they have different colors, they have different sizes. Some are short, some are thin, some are stout, but we consider them belonging to the same species. Now, if these organisms belong to the same species, then why do they look different? You know why? The answer for that is evolution. So, what is evolution? Evolution is a process that results in changes in the genetic material of a population over time. Evolution reflects the adaptations of organisms to their changing environments that can result in altered genes, novel traits, and even new species. Now, your evolutionary processes depend on both changes in the genetic variability and changes in the allele frequencies over time. Now, we have here a balanced rocks or stones. Now, I have a question. Can these stones keep balance indefinitely? Yes, they can maintain balance or equilibrium if there is no outside forces that influences your stones. Now, if we are to compare these stones into a population of animals like this one on the screen, can equilibrium also be maintained within a population's genes? The answer for that would also be yes. The equilibrium may also be maintained within a population's genes if there are no external factors affecting the population, meaning there is no evolution occurring in that population. Now, how can we say that an evolution will occur in a population? Now, evolution will not cannot occur in a single individual alone because evolution is a process of changing the gene frequencies within the gene pool. So therefore you need to have indefinite number of individuals for in order for you to see evolution happening in the allele frequencies or the genotypic frequencies. Now, we have different forces that may cause genetic variation and evolution in a population. So you have natural selection, you have genetic drift, you have genetic hitchhiking, and gene flow. So you already know what are those things. Now, lastly, how can we test whether evolution is occurring in a population? So when can we say that there is an evolution occurring? When in fact evolution cannot be easily be identified within a population. Some evolution may be on the genes, alteration in the genes, which will not be sometimes will not be um, phenotypically present. So with that, scientists may use a principle called the Hardy-Weinberg Equilibrium to check if there is evolution that is happening or occurring in a certain population. So for today, we'll be talking about the different mechanisms that produce change in populations, specifically your Hardy-Weinberg Equilibrium. Now, what is Hardy-Weinberg Equilibrium? So you may think that what is Hardy-Weinberg? I'm not too familiar with that. So this Hardy-Weinberg equilibrium, it describes the population that is not evolving. So meaning there is no evolution that is happening in a population. So The frequencies of alleles or gene versions and genotypes or sets of alleles in that population will stay the same over generation. So it may be from the parental to the filial or until the fifth generation, the frequencies of the alleles and the genotypes will all be the same or equal. So if a population and so what will happen if a population does not meet the criteria of the Hardy-Weinberg principle? So what do you think will happen? So if there since Hardy-Weinberg equilibrium will maintain the same genetic frequencies in the alleles and the genotype over a number of generations, and we can say that when we will not meet the criteria, then therefore there is a change that is happening in a population. So therefore evolution is occurring within the population. Now let's take into an example, these two beetles here. So suppose there is a large population of beetles and then it comes into colors. You have the dark colored beetles and the light colored beetles. Now, the colors are determined by the A gene having two alleles of a locus. Question: what will be the phenotypes and genotypes of the individuals of the population? So, considering the dark gray will be is dominant over the light colored beetles. So, the answer for that is the dark colored beetles will have a phenotype of homozygous dominant A and heterozygous, while your light colored beetle will have a homozygous recessive genotype. Now, in the population, suppose that the A allele, the capital A allele, has a frequency of 0.3, while the allele A, small a, has a frequency of 0.7. So we can assign that the allele frequency for the dominant allele A will be 0.3. So it may be found in the dark colored beetles, while the recessive allele will be found on the light colored beetles. Now, notice that when you add this frequency 0.3 and 0.1, it is equal to 1. And if we represent that 1 into percentage, you can also see that it will be equal to 100%. So in the Hardy-Weinberg equilibrium, so the population is always equal to 100% or 1. Now, so what are the assumptions to be considered for a population to be in Hardy-Weinberg equilibrium? So when can we say that a population is not evolving? So to be in a Hardy-Weinberg equilibrium, we need to consider 5 assumptions. Okay, so what are these assumptions? So we have first, there is no natural selection. So all alleles confer equal fitness, making organisms equally likely to survive and reproduce. So let's take an example, this different colored frogs on the screen. So you have here dark green frogs and light green frogs. So if they are in nature, if there are predators present in the vicinity, the light green frogs would definitely be the first prey for the predators. Why? Because they have light colors and they could easily be seen. So your dark colored frogs will have higher survival rate than those of the light green frogs, and they can reproduce a high rate of reproduction because they will be a lot in the population as compared into the light green frogs. Now, in the Hardy-Weinberg population, there is no such thing as all the organisms have equal fitness. So regardless of their color or their other characteristics, though, they have equal rates of survival and reproduction. So the second assumption that we have is there is no mutation. So no new alleles are generated by mutation, nor are genes duplicated or deleted. So the genes and the alleles should all be the same throughout how many generations. So there should be no mutation. So even minute duplication or deletion in the genes should not be observed in a Hardy-Weinberg equilibrium. Next, there should be no migration. So neither individuals nor their gametes enter or exit a population. So take an example, these frogs here, you have two populations. They are divided, they are isolated by their habitat. So you have a river there. So the other population is on the other side of the river. Now, in Hardy-Weinberg equilibrium, the frogs could not pass the barrier. So there would be no interbreeding with the different populations. So the organisms within the population will never migrate to the other population. Next, the fourth assumption is we have the large population. So the population should be effectively infinite in size. So again, in order to observe evolution, we need to have an indefinite number of organisms. So if we only have a few organisms then we could not tell that evolution is occurring. And the last assumption is we have random mating. So the organisms mate randomly with each other um, with no difference for particular genotypes. So regardless of the colors or the characteristics, then the animals or the organisms would mate randomly. So in nature, it is not observed because when organisms mate, they try to look for partners that can pass good genes into the next generation. So in here we have random mating. So everyone has an equal chance to mate with other organisms. Now, since we all have that assumptions, what will happen if any of those assumption is not met in a population? So what do you think will happen? So if even one assumption is not met in the population, it will not be in Hardy-Weinberg equilibrium. So what do you think will happen if it is not undergoing Hardy-Weinberg equilibrium? Then the population would definitely be evolving. So the allele frequencies may change from one generation to next. Allele and the genotype frequencies within a single generation may also fail to satisfy the Hardy-Weinberg equation. Now, in real life, does Hardy-Weinberg equilibrium generally happen? So, so there no natural selection in the environment, no mutation? Can we observe that one in nature? The answer is no. Now, so if Hardy-Weinberg equilibrium is not realistic in nature, then why does it matter? Why is it important to study the Hardy-Weinberg equilibrium? Okay, so it is important to study the Hardy-Weinberg equilibrium because it gives us the baseline to compare how an evolving population is as compared to a population that is not evolving. So we using the Hardy-Weinberg equilibrium, we can see that a population is evolving or not. Now, if a population is in Hardy-Weinberg equilibrium, the allele frequencies will be related to the genotype frequencies by a specific mathematical relationship. And that is the Hardy-Weinberg equation. Now, the Hardy-Weinberg equation, you have two equations to be used. So we have P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. So it's biology, but why do we have math here? So here we're trying to compute the frequencies of alleles and the genotypes of the population. Now, what do we mean by those P's and Q's? So let's have the first equation. So P plus Q equals 1. In this equation, P is dominant allele frequency in the population, while the Q there, it represents the recessive allele frequency. So this equation is used to identify the allele frequencies in the population. Now, take note that the P doesn't always have to be equal to Q. And the P's and the Q's there can have varied numbers. So it doesn't have to be equal, and it doesn't have to be a whole number. Now, the dominant allele, which is P, doesn't always have to be larger than the Q. In some instances, the recessive allele may also be larger as compared to the dominant allele in the population. Now let's go to the second equation. So you have here P squared plus 2PQ plus Q squared equals 1. So the P squared there in the equation is the homozygous dominant frequency, while the 2PQ represents the heterozygous frequency, and the Q squared is the homozygous recessive frequency. So now we have all the equations. You already know what are different assumptions to consider to know if a population is undergoing Hardy-Weinberg equilibrium. Now, um, for you to further understand the Hardy-Weinberg equilibrium, so let's have an activity. So for this activity, you should prepare this material. So you should have 2 large bags of milk-coated fish-shaped biscuits. So it may be knickknacks, or you can use pretzels since they have different colors or flavors. You could also have 2 large bags of chocolate-coated fish-shaped biscuits, or your knickknacks or your pretzels, your plates or napkins, and a large bowl where you will place your food. Now, the major preparations and procedures. So you need to pour the contents of all the bags of the fish-shaped biscuits into a large bowl. So this bowl will represent the leg. Now, you are tasked um, to consider that the dark brown or the chocolate coated biscuits are the homozygous recessive individuals. So meaning if we use letter G as our allele, so it will have all small g's. And the white milk coated biscuits displays the dominant phenotype, and therefore may be either homozygous dominant G or heterozygous individual. Now we have 2 activities to be performed. So in part A, part 1, activity 1, you'll need to perform the perform the activity to check the Hardy-Weinberg equilibrium. And the second is you need to compare the Hardy-Weinberg equilibrium and natural selection. Now, for the specific procedures of these activities, you can check the link on the description box. Okay. Now, you can pause this video and take your time and follow the procedures and directions. And after we will discuss the results of your data collection. Okay, so once you have collected your data, you can now compare the genotype frequencies in both simulations. So the first simulation is in the Hardy-Weinberg equilibrium, and the second simulation is the one where you compared the natural selection and the Hardy-Weinberg equilibrium. Now, in the first simulation, your results should have a fairly consistent frequencies over the 5 generations. Why? Because there should be no evolution that will occur in your first simulation, because it is undergoing Hardy-Weinberg equilibrium. When you introduce natural selection, then you could see that the genotype frequencies will be varied over the 5 generations. So there will be differences. With that results, you can see that evolution is occurring in the second simulation because over the 5 generations, you can see differences over the genotype frequencies. Now, um, in order for the frequencies to maintain stable over multiple generations, then you can go back to the different assumptions. So again, no natural selection, no mutation, random mating, no migrating, and also, and the last one, I forgot. Okay, now you have already um, known the different equations and the different um, assumptions to consider in a Hardy-Weinberg equilibrium. Now, let us try to apply the equations that we have tackled a while ago. So, let's do problem solving. So, let's have here the first problem. So, a population of frogs is in Hardy-Weinberg equilibrium. So, there are a total of 500 frogs and 375 of those are dark green, while the rest are light green. So let us consider that the allele for this population is G. Now, we need to answer these specific questions. What are the genotype frequencies and what are the allele frequencies? So let's have a step-by-step process in solving this problem. So the first thing you need to do is to determine the equation to work with first. So remember, you have two equations. You have the p plus q equals 1, p squared plus 2pq plus q squared equals 1. Okay, so you need to choose the equation that is suitable for your equation. So what equation will you use will depend on the given data on your problem. So for this problem, since we're dealing with individuals, then we'll be using the second equation, which is the p squared plus 2pq plus q squared equals 1. Okay. Now let's go to the second step. So the second step, you need to figure out the values that can be determined in the problem. So you have the data, the total number of frogs is 500. And you also have the number of dark green frogs, which is 375. And for the light green frogs, the data is unknown. So the first thing you should do is to look for the number of light green frogs. So how should you do that? So 375 plus x equals 500. Now, to find for x, 500 minus 375, and that would be x is equals to 125. So therefore, there are 125 light green frogs. Now, the data that we have obtained is still insufficient. So we need to find the frequencies of the data using the this equation. So let's find the frequency of dark green frogs. So you have the data 375 divided by 500 total number of frogs, and that would be 0.75. But we know that 0.75 is the frequency of dark green frogs. So therefore, it may either be homozygous, dominant, or heterozygous. So this data that we have here cannot be used. So let's go to the second data. So we need to find the frequency of the light green frogs. So how to solve for that is you need to divide the number of light green frogs over the total number of frogs. So that's 125 divided by 500, and that would be equal to 0.25. Now, we all know that 0.25 for the light green frogs are all homozygous recessive. Now, this data can be used. Now, since those are so therefore that 0.25 frequency, genotype frequency is the Q squared, since we all know that Q is the recessive. So we need to identify the other values using the data on the Q squared. So what we should do is to supply the values that is known. So P squared plus 2PQ plus 0.25 equals 1. Now we need to find the P squared and the 2PQ. So how to find for that is you need to substitute the different values. Now let's go to the third step. So in the third step, you already have the value of Q squared. So you need to take that value and then calculate the remaining values. So Q squared, so since you have Q there, we can identify Q squared, we can identify now the Q. So what will you do is to get the square root of Q squared. So Q is equals to square root of 0.25, and the answer for that is 0.5. So therefore Q is 0.5. You have now the value for Q, so you can now find for the value of P. So how to look for the value of P? So let's put the given first, 0.5, and then P is unknown. So you will use the formula P, the first equation, P plus Q is equals to 1. So P is unknown plus 0.5 equals 1. So P is equals to 1 minus 0.5, and P is equals to 0.5. So we have now the values for P. Since we have the values for P and Q, we can now solve for the P squared and the 2PQ. So again, let's write the given. We have the P 0.5. Your Q squared is 0.25, and your 2PQ is unknown. Now, let's try to put the equation. So P squared plus 2PQ plus Q squared equals to 1. Now, substitute the values. So 0.5 squared plus 2PQ plus 0.25. So we use 0.25 because we already know the value for Q squared. So substitute the other values. So for P squared, that would be 0.25 plus 2 times 0.5 times 0.5. Plus 0.25 equals 1. Now, so the final answer would be the P squared will be 0.25, and your 2PQ would be 0.5, and your Q squared is 0.25. Now, let's have the final answers. Okay, so for the final answers, we have here, for the question A, you are looking for the genotype frequencies. So P squared, you know the values of P squared, that is 0.25. 2PQ is 0.5. Your Q squared is 0.25. Your P squared there represents the homozygous dominant. Let's have the values of P, 0.5. Values of Q is also 0.5. Now, the P square there, it represents the homozygous dominant genotype, while the 2PQ represents the heterozygous genotype, and your Q squared represents as dominant recessive genotype, homozygous recessive genotype, and your P and Q are the alleles. So that is the final answer. So do we have the same answers? Okay, if in order to check if your answers are all correct, you can check it by adding all the data and see if it adds up to 1. So if it all adds up to 1, then your answer is correct. Okay, so let's try to apply the things that you have learned through our step-by-step problem solving. So let's apply that one to this practice problem. So the first problem, a population of infants in Tanzania was scored for the beta hemoglobin locus genotype that is determined by a single autosomal locus with alleles A and S as follows. So your homozygous A has the number of individuals are 189. Your heterozygous individuals are 89 in the population, and your homozygous S individuals are 9 in the population, having a total of 287 individuals. Now, as you can see, not all the time the number of individuals are in a whole number. So for this example, you can see that there are only 287 individuals in the population. Now, we need to answer the specific questions, characterize the gene pool by the allele frequencies for A and S, and B, predict the genotypic frequencies. Now, the first step is we need to identify the equation. Now, you can pause this video and try to solve, and let's see if we have the same answers. Okay, so have you chosen the equation? So the equation that we'll be using is the second equation, and that is p squared plus 2pq plus q squared equals 1. Why? Why do you think so? It is because the given data in the problem is and the number of individuals. So the first thing we should do is to look for the frequencies of different individuals. So we have here the data for the genotype, the number of individuals in a specific genotype. So we have here the data or the individuals that having the characteristic is having codominance. So having A and S will determine that an individual is heterozygous. Okay, so what should we do first? So we need to let's go to the answers. We need to predict first the genotypic frequencies because we have the number of individuals for each genotype. So it's easier for us to identify the frequency. So in order to identify the frequency, so let us consider the homozygous dominant A as p squared. Why? Because they are abundant in there are a lot of people and that having that genotype. And let's consider AS since it's having the two alleles, so it's heterozygous. And then the SS is homozygous S, we'll consider that one as recessive or your Q squared. Now, to identify the frequency of your P squared, so you need to divide 189 individuals over the total number, which is 287. The answer for that would be 0.66. Now, remember, if it is in frequency, your answer should be in a decimal, should be in decimals. And if it is in percentage, it should be in percent. Now, you have already your p squared, and that is 0.66. Now, let's go to q squared. You need to divide 9 from 287, and that would be 0.03. And 2pq, since there are 89 individuals, you need to divide it to 287. You have now 0.31. Now, you have all this data. Now, we can now solve for the allele frequency. Now, if you have noticed from our previous example, we identified first the allele frequency. It is because in this problem, we already know the heterozygous frequency. From the previous problem, it is still vague because you are only given the data that the dark green frogs are, the dark green frogs may be hetero or maybe homozygous dominant. Okay, now to solve for the allele frequencies, you need only to square root the values for the p squared and q squared. So since p squared is for the homozygous A, so therefore the p is A. So if you square root the answer of 0.66, your answer would be 0.81. And for the s, so square root of q squared, that is square root of 0.03, the answer is 0.18. So, do we have the same answers? If you have, then let us check if our answers are correct. So, to check that one, you need to add up all the data for the genotypic frequency and the allele frequency and check if it adds up to 1. So, are we good? Now, let's have another example. So, another practice problem. So, a population of sheep is in Hardy-Weinberg Equilibrium. So, the allele for white wool, capital W, has an allele frequency of 0.19. And the allele for black wool, for lowercase w, has an allele frequency of 0.81. As you can see in this problem, the data that is given to you is in frequency. So you don't need to solve for the allele frequency because it's already given in the problem. Now, since the white wool has a uppercase W, then we can consider that one as the dominant allele, while the lowercase w, we can consider that one as the recessive allele. Now, we need to find the the percentage of heterozygous individuals and the percentage of homozygous, dominant, and recessive individuals. Now you can pause this video and try to solve for the percentage of individuals. Okay, so let's try to check your answers. Okay, so we have here, since you already know that allelic frequency, so we should directly go to the genotype frequency. So we'll still be using the second equation, and that is p squared plus 2pq plus q squared equals 1. Now, so to solve for p squared, so I'll solve p squared first. Or shall we have 2pq? Okay, let's have 2pq since it's in question A. So in 2pq, no, you need to multiply 2 and the values for p and q. So you know that the p is equal to 0.19 and q is equal to 0.81. So the answer for that is 0.31. Is that the final answer? Yeah, it is not the final answer since we're looking for the percentage. So you need to multiply that 1 into 100, and the answer would be 0.31 times 100 is equals to 31. So there's 31% of individuals having heterozygous genotype. Now let's go to question B, the percentage of homozygous dominant. So you need to square the value of P. So P squared. So P is 0.19 squared, and that is 0.0361. That is not the final answer since we're looking for the percentage. The answer would be 3.61%. And that is the percent of homozygous and the homozygous dominant individuals in a population. And the last is the homozygous recessive. You have their Q. So you need to square Q. Q is 0.81 squared and that would be 0.6561, and multiply that 1 into 100 to find the percentage, then you have 65.61%. So that is the number, the percentage of individuals having homozygous recessive genotype. Now check your answers if you can see if it adds up to 1. Now, if your answers are 0.98 or 0.99, please consider that you have rounded off the values. before the final answer. So there are times that it will not be always equal to 1 because you have rounded it off. Okay? So I guess you have really understand now the Hardy-Weinberg equilibrium, its uses, and how to solve for the frequencies of alleles and genotype in a population using the Hardy-Weinberg equation. Now, if you have any questions, you can comment down below and I'll try to answer it. See you in the next video, everyone.